get started then. So first of all, thanks everybody for coming to the seminar series. Today we have Gordon MacLeod, who's going to share with us his proposal presentation that is very, very interesting and unique for the agronomy department. And it's looking at the effect of scarab beetle larva activity on soil properties. And so with that, I'm going to turn it over to Gordon. Gordon, you can go ahead and share your screen. And I just ask that everybody keeps their mics muted throughout the presentation until afterwards.
All right. So, yeah, thanks, Rick. Thanks, everybody, for tuning in. My name is Gordon MacLeod. I am a PhD student in the agronomy department. And my major advisor is Dr. Tim Filley of Eepson Agronomy. I'm co-advised by Dr. Douglas Richmond of entomology. So this is really an interdisciplinary project between agronomy and entomology, and it's funded by a grant from the USDA NIFA. Today I'm going to be going over my proposed research, which is the effects of invasive scarab larval activity on soil, chemical, and physical properties. So an overview of the presentation, I'm going to talk briefly about soil ecology of invasive invertebrates, why we're looking at the Japanese beetle, and then some project objectives, project components, and then methods we're using to address those objectives. This presentation is primarily going to be about methodology of the project, but I am in my third year, so I'm beginning to work through some data analysis at this point. So I'll give a brief overview of where the project stands, and then there will be time at the end for questions and comments. So there are over 2,000 recognized invasive invertebrate species in the continental US. And by definition, an invasive species does harm to its environment, usually by monopolizing or manipulating resources. So what tends to happen is that they can outcompete native species for resources, especially in the herbivore category, which is what we're looking at. In soil-dwelling species, the effects that we tend to see over time are that they alter carbon and nitrogen dynamics and storage, and as well as water dynamics in the soil. In the case of these lumbersig earthworms on the left, many studies have been done looking at carbon and nitrogen dynamics in the soil with them, and found that in general there's a net loss of organic matter from the soil, or rapid incorporation of organic matter into the soil, which results in mineralization of carbon and nitrogen. And over time, you can see macroflora population changes associated with their presence or their introduction to a new area. Burrowing and mining of soil-dwelling invertebrates also changes considerably, as well as direct carbon inputs to the soil, as I mentioned already. These Formosan termites in the middle are a good example of that. And then also a characteristic shared by the Japanese beetle, which is our subject invertebrate with this Argentine ant on the right, or that they can proliferate non-host plants and other pests either directly or by association indirectly. So most recent estimates say that $17 billion is spent annually on control and mitigation of invasive invertebrates in the US, and over $2 billion of that is spent on insecticides alone. So this is a major thing, and many of these are particular to certain areas of the US, but some, as in the case of the lumbersig earthworms, are widespread. Looking at the Japanese beetle specifically, we're studying this primarily because it has several analogous behaviors to other invasive soil-dwelling invertebrates that I mentioned on the previous slide, particularly the lumbersid earthworms. And as you can see from the estimated distribution map on the right, over an 80-year period, their infestation area has increased pretty rapidly. Over about an 80-year period, they've spread from the east coast of the US, where they were introduced around the early 20th century, all the way to the Midwest and up into Canada, as well as some parts of the Southeast. So this is the current distribution shown in orange, and it's updated within the last 10 years or so. So as you can see, it's been spreading westward across the Great Plains, with some pockets popping up in the western states, primarily around city centers where there are airports or freight terminals where they can catch an easy ride. According to the USDA APHIS, the species has established populations in at least 36 states and 5 provinces, and that over $460 million is spent annually to control Japanese beetle infestations, with over 234 million of that spent on the larval stage alone. So the beetle has a 1-year life cycle. The adults emerge in early summer, and per individual, they tend to live an average of 45 days. Although the period of emergence is generally the entire summer, is when you'll see the adults out. During this time, they're eating, they prefer to eat the soft leaf tissue between veins. So this is a common site where you'll see skeletonized leaves in an area where they have been feeding, essentially defoliating plants. And they tend to aggregate at food sources due to pheromone attraction. So in localized areas, you'll see swaths that they've just completely eaten bare. There are over 300 known host plants, including several agricultural species, many orchard trees. Corn and soy are near the top of the list, as well as ornamentals, pastureland grasses, and turf grasses. And during the summer period, the females will also continually burrow into the soil, or I should say repeatedly, as many as 20 times over the course of their adulthood, and each time lay anywhere from 16 to 20 eggs per clutch. So they proliferate quite rapidly. After about a 2-week embryonic development period, the larvae hatch, and they mine through the soil for the next 9 to 10 months. So they overwinter in the soil, and we're really mostly interested in this life stage of the beetle, primarily because it's the longest and it's the least studied. Most of the research that's been done so far has been regarding physiology and chemical susceptibility of the larval stage. So we do know some things. They prefer to eat plant roots, and they prefer live plant roots to soil organic matter, although they can survive on soil organic matter alone. They have an alkaline midgut, and the midgut can be as high or higher than 9.5 on the pH scale. And then recently, there's been an ammonia nitrifying bacteria found in the midgut, and this is part of the work that's been done by Dr. Evia Arias and Dr. Richmond's lab over the last couple of years. So these are some major considerations that we are bringing to bear in the current study, but largely their impact on their soil environment is unknown. So we're breaking down these knowledge gaps into 3 categories. The first is their effect on physical properties in the soil, their effect on those aspects of carbon and nitrogen cycling that are in the soil, and then also to better characterize their feeding strategy. So an overview of our objectives intended to address these knowledge gaps. First, we're looking at the effect of larval activity on soil physical properties. Second, on carbon and nitrogen stocks, and specifically interested in bioavailability of that carbon and nitrogen. And then third, looking to better characterize their feeding strategy or feeding preferences. So essentially, the first two objectives are impact-based, and the third is a process-based objective. I'll put these on the side as a reference going forward. So with the first objective, we hypothesize that larval root herbivory and soil mining will alter soil physical properties, and specifically that soil will have greater hydraulic conductivity due to preferential flow, and that soil aggregates will have a smaller mean weight diameter relative to non-infested soil. With the second objective, that the gut provides an environment that facilitates incorporation of organic matter into soil aggregates and onto clay mineral surfaces, and that both of these will increase. Also with the second objective, that increased nitrogen cycling will increase mineralization of nitrogen, and that this will result in a net loss of nitrogen due to leaching. And with objective 3, that larvae will exhibit a root branch order feeding preference according to food quality. And by food quality, I mean that they will prefer root branch orders with a lower carbon and nitrogen ratio. So the project is broken down into 3 components or experimental setups. The first is a set of cylinder mesocosms that were installed at the Throckmorton Purdue Agricultural Center. And these are intended to address objectives 1 and 2, looking at physical and chemical changes in the soil itself. The second are a set of lysimeter mesocosms that were also set up at TPAC, also intended to address objectives 1 and 2, looking at nutrient leaching and moisture. And the third are a suite of feeding assays using live grubs and likely food sources, intended to address objective 3, primarily looking at feeding strategy and preference, but also looking at direct inputs of carbon and nitrogen to the soil from their frass or their fecal matter. So the methods we're using to address these objectives, we're doing wet sieving of soil samples from the cylinder mesocosms to look at aggregate stability, and then also doing isotope ratios of soil samples from the rhizosphere and deeper soil, and applying a 2N member mixing model to look for potential increased soil mixing in an infested system. And then also with the lysimeters, we have volumetric moisture sensors installed in the soil column, as well as water level probes to be installed in the water reservoir, looking at water dynamics. With objective 2, density flotation of bulk soil samples from the cylinders to look for, by sodium polytungstate, to look for organic matter association with minerals, and then and also performing elemental analysis on those fractions. Elemental analysis and isotope ratios of soil size fractions from the aggregate stability analysis, looking for carbon and nitrogen incorporation into aggregate structures, addressing that physical protection component. And then also amino sugars analysis we're using as a proxy for microbial biomass accumulation or microbial necromass. With the lysimeter water, we're doing ion chromatography, specifically interested in nitrogenous nutrients, but also we will be receiving additional cation and anion data that can be brought to bear, and then total carbon of lysimeter water that's also been filtered, so looking at dissolved carbon. With the feeding assays, again, looking at direct organic matter inputs to the soil from the frass by elemental analysis. And then also for objective 3, doing thermochemolysis of frass samples as well as root branch orders, and looking for similarities across samples of biopolymer ratios to address the feeding strategy question. So a little bit of background about the experiment setup. The open-ended cylinders were installed in fall of 2018, and they were left in the field to allow the soil to equilibrate over the winter. They were seeded with Kentucky bluegrass in the spring, which is a favored oviposition plant for the female adult, and treated in summer 2019, and then again this past summer. There are 9 replicates with 4 plots per replicate and a complete randomized complete block design. And the sampling matrix is 2 treatments of infested or not infested by 2 incubation lengths, 1/2 and 1 1/2 life cycles of the beetle. There are 3 cylinders per plot as shown in the diagram, and all 3 cylinders per plot receive the same treatment. So the mean value of these cylinders is used as the single value per plot for that replicate. With the lysimeter mesocosms, these were installed this past summer, and the intent is to leave them in for multiple life cycles of the beetle, at least 1 life cycle, but preferably 2. They were treated this past summer, and the intent again is to infest them again next summer. There are 16 of them with 2 treatment levels, again infested and non-infested, and they were randomized by treatment. And they're sampled after rainfall accumulation of 2 centimeters or more. For the feeding assays, there are 2 of them at present. The first is looking at feeding preference by branch order. So we take live grubs from an infested monoculture. This is a turf grass farm that has been infested with beetle grubs. And we're looking at a comparison of what they were, or their frass, to their likely food source. So we take the grubs directly from the soil, clean them off, and put them into glass scintillation vials, allow them to void their gut contents. And then we collect that frass as an example of what they were eating directly before we remove them. Then we also take the roots from that monoculture and dissect it by branch order, excuse me, as well as some of the soil from that location, and analyze all of these samples separately by pyrolysis, gas chromatography, mass spectrometry, and elemental analysis and isotope ratio mass spectrometry. And then these results are compared for similarities. Here's an example chromatogram of that thermochemolysis by pyrolysis. So on the x-axis, there's retention time. On the y-axis is total ion count. So essentially each peak is representative of the abundance of that compound as it elutes off of the gas chromatography column. So the black line is a larval frass sample. The pink line is a 3rd branch order roots, and the blue line is a 1st branch order root. And already you can see that there are some differences in abundance of these compounds, but generally we see the same compounds popping up across the chromatogram. And we can break them up into 3 categories. So the first is a series of sugar fragments. The second are a group of aromatic compounds, primarily in the cinnamic acid family. And then the third are aliphatic compounds shown on the right, eluting later in the chromatogram. So these second and third groups we're using as proxies for lignans and tannins and suberins, respectively. And assuming that their structure is conserved through gut passage, that we haven't missed any bacteria in analysis that are able to break these down, that we can use the ratios of these 2 groups, these 2 latter groups, as indicators of biopolymers from their food source. We're doing a similar analysis to look at gut transit time, or the time it takes for their food material to pass through their gut, and then potentially down the road have an idea of soil turnover rates and organic matter turnover, except that the setup for this is a little bit different. We take the grubs and allow them to feed only on soil organic matter from a different prepared soil for 2 weeks. And then we take them from that soil and allow them only to eat 2nd branch order grass roots for 4 days. And we collect the frass from those vials every 8 hours. So essentially what we end up with is a chronosequence of the contents of their gut over a 4-day period at 8-hour increments. And then we analyze those samples in the same way as mentioned before. So as I mentioned earlier, I'm in the 3rd year of the project. So as an overview of where it stands currently, the first year was essentially spent in lit review and pilot data collection. The figure on the upper right is an example of percent carbon analysis that was done on samples from an experimental setup by Dr. Evia Arias, who's again the postdoc on Dr. Richmond's project. She had this separated by, or she had her infestation levels at a control or uninfested level, and then low and high infestation levels. But we found actually that with population densities in the mesocosms after they were deconstructed, that there was no real statistical significance between the low and high infestation levels. So that informed my experimental setup, and I grouped those together, attempting to infest each infested treatment level as a single infestation level. And then my control is the not infested treatment level. The graphs on the bottom are examples of feeding preference assays that we were doing, or feeding assays. So the one on the left is a proportion of compound group by sample, and the compound groups again are those sugar, aromatic, and aliphatic compound groups across branch orders and larval frass samples. And then the one on the right is looking at percent carbon and nitrogen in those same samples, as well as the carbon and nitrogen ratio. And what we see in the pilot data, at least, is what we expected to see, that sugars are depleted in the frass compared to branch order roots, and also that ratios of aromatic to aliphatic compounds are more similar to smaller branch order roots with a lower carbon and nitrogen ratio. The experiment setup was essentially the 2nd year of the project, so I won't go too much into this unless you have questions about it at the end, simply because we went over most of it previously. And then I'm currently working on sample analysis and data analysis for that first sample set last fall. Still working through carbon and nitrogen data, as well as this is an example of the aggregate stability test for that rhizosphere. So on the x-axis, again, size fraction from the soil, and then on the y-axis is the mean difference between infested soil in that size fraction and uninfested soil. So ultimately, in the first round, again, this is after 4 months of infestation or larval activity, we don't see statistical significance at a 95% confidence interval between any two groups, but we do start to see some separation between groups. It'll be interesting looking at this next sample set that we actually just took out of the ground last week after having another year of larval infestation. Here's an example of the first 2 sample sets from the lysimeter water run by ion chromatography. There are a lot of caveats to this. The soil was heavily manipulated and during setup, and the purpose for that was to ensure as little variance as possible, or the most similar setup across all lysimeters. However, the downside to that, at least immediately, is that I'm sure we disturbed a lot of things that are now flushing out of the soil. So during this first few months, we're probably seeing a lot of noise. I don't want to put too much stock into this until we've seen the long-term effects of these leachates. So really, I'm taking a long-term perspective on this, and I hope to have an update for you in another few months. But already we start to see a little bit of separation with nitrates and nitrites between treatment groups. So that's something to keep an eye on going forward. So I'd like to acknowledge my advisors, Dr. Philly and Dr. Richmond, my committee members, Drs. Bruder and Dr. Gonzalez, as well as several people who have given me advice and helped me with analysis. Dr. Avia Arias, Dr. Sparks, Dr. Kladivko, the folks at the National Soil Erosion Lab, especially Brenda Hoffman, and then my classmates and colleagues in the Philly lab, the Richmond lab, and the Purdue Stable Isotope Lab. And with that, I will open it up to questions and comments.
Questions for Gordon? If you have questions, remember to unmute. Okay. What level of like infestation do you have in the mesocosms? Like, I guess, what's the density of beetles in the mesocosms that you have?
So, yeah, we don't actually know until we deconstruct the soil. So, what we've been seeing is anywhere from 3 or 4 on the low end to 30 on the high end. So really within our infested treatment group, we do tend to see kind of a good regression of grub densities across cylinders. The way that we infest them, if this is what you're asking, is to put equal numbers of female and male adult beetles into the cylinder and then cage them in there. So we leave them there for 2 weeks and allow them to eat and propagate. During that time, the females are burrowing into the soil. We also cage the control plots so that no beetles can get in. We don't get a natural infestation where we don't want one. And then after a week or 2, we remove the cage and do it again. So essentially, we're not one problem that we had with other studies is that if you add grubs directly to the system, you may end up with a higher than natural infestation density. So one thing that we're hoping to achieve by this method, and this is a pretty common method actually, is to essentially allow the infestation density to be more indicative of what you would see in a natural setting. So yeah, just by virtue of the amount of volume of soil, the idea is that the beetles aren't going to infest at a higher density than would be than you would see elsewhere.
Okay. And then my other question that I have is, how much disturbance is happening to the soil in the cylinders when you go out there and sample? So I guess what I mean is, you're using the same locations each year, right? And so how do you maintain the integrity of of the soil within the cylinder when you're taking samples from it?
Sure. So there's two experiment setups. I guess I'll pull up this slide again and we'll see. I'm not quite sure. I'm sure I know what you're asking, but so with these, are you talking about the cylinder mesocosms or the lysimeters?
The cylinder ones.
Okay. So with these, we're only sampling half of them at a time, and we don't put more cylinders in, or I haven't put more cylinders in after cylinders were removed. So whatever was there in that spot where the cylinder was infested or not infested is removed, and then uh, the following year or this past year, the remainder of the cylinders were removed. So we're not repeating the experiment in the same location, if that makes sense.
Okay. Yeah, that's what I was asking. So,
Okay.
okay. Interesting. Other questions for Gordon?
Thinking about Seth's presentation, I think I'm getting off easy.
Gordon, can I ask you a question?
Sure.
So if I remember correctly from class, however many years ago that was, maybe 2 years ago, and so I might not remember correctly.
Yeah.
I think one of the questions that we started out with asking was if you would see changes to carbon and nitrogen in just the time frame of like your thesis. So far from what you're seeing, are you surprised by results, or is this what you're expecting, or do I even have that question wrong and that was not one of the questions that we had for you?
Yeah, no, if I remember correctly, that was a concern brought up in that class and one that we've discussed in our own lab quite a bit. So what we are attempting to look for here are not necessarily changes in the more recalcitrant forms of carbon, but rather looking at incorporation of carbon into aggregate structures, which can form over a shorter period of time, or mineralization of the more labile forms of carbon and nitrogen. And an example of that are leaching of nitrates, ammonium, leaching of or incorporation of carbon into microbial biomass and changes in that potentially. Yeah, and then with the biopolymers, we recognize that a lot of those aren't going to change over such a short time. So, and that's why actually we're using them as indicators of feeding preference, because they're much less likely to degrade over such a short time. Does that make sense?
Yeah, that does make sense. Thanks a lot, Gordon. Great presentation.
Anybody else with questions for Gordon? What method are you using to measure your dissolved organic carbon?
It's a Thermalox TOC machine. So it combusts the organic material into CO2 with a continuous flow of oxygen. And then the CO2 is measured as the metric for total carbon in that sample.
Okay. Anybody else? Okay. Hearing none, I think you got off a little bit easy, Gordon.
Yeah, no kidding.
But hearing none, we will go ahead and finish with there. Next week, we have a presentation on some different starter fertilizers in corn. So join us for that. And with that, let's give Gordon a virtual round of applause because that's all we can do right now. And everybody have a good weekend.
Thanks, everyone.
Thank you. Good job.